Okay, so what I want to do now is talk about some skeletal muscle anatomy. We'll review some gross anatomy here, but eventually we're going to get down into the microscopic anatomy, look within the muscle cell here, and look at some of the details of that, because that's going to be critical for you guys to understand how skeletal muscle contraction actually occurs. Okay, so let's start gross anatomy though. Here's our skeletal muscle right here. And, you know, there's connective tissue sheaths around the skeletal muscle. The sheath that wraps around the entire skeletal muscle is called the epimysium here. And then you can see that the skeletal muscle is attached to bones via tendons. Okay. So we take a cross-section of the skeletal muscle here. And what you see all these individual new cross-sections. And these individual new cross-sections, which is what we're looking at right here, are called fascicles. So each of these individual guys are called fascicles. Now, if we look within a fascicle, you can see that there's other cross-sections. And this next level of cross-sections that we're dealing with right here, those are actual skeletal muscle cells. Those are muscle fibers, skeletal muscle cells. So this is one muscle cell right here. And one of the reasons that's important, why I want you to know that, is because when motor neurons come down and act on skeletal muscles, cause them to contract, the level that they're innervating are the individual skeletal muscle cells. They don't come down and cause the whole muscle to contract. They don't come down and cause the whole fascicle to contract. That doesn't happen. What happens is these motor neurons come down and they cause the individual muscle cells to contract. And what that does is it allows us to have a much greater control over muscle contraction. So skeletal muscle that contains fascicles, fascicles that contain skeletal muscle cells, and then further, if you look at the skeletal muscle cell, you can see these new structures, and these new structures here are called myofibrils. They're these rod-like filaments that run the length of the skeletal muscle cell. And those are called myofibrils. And that's where we're going. Okay. So, a single muscle cell contains hundreds to thousands of densely packed myofibrils. These are rod-like structures that run parallel to its length. So there's your skeletal muscle cell, all these mitochondria in there, right? Need all those mitochondria. Well, why do we need those? What are those going to produce? Those are going to produce ATP. Okay. So we have a lot of mitochondria in there. Pause it real quick. Okay, so yeah, here's our muscle cell here. And the muscle cell contains hundreds to thousands of these densely packed myofibrils, and these are running parallel to the length of the muscle cell. Now let's take a close-up look at one of these myofibrils. And myofibrils contain an important feature. Myofibrils contain these structures called sarcomeres. And this, from right there to right there, is a sarcomere. From right here to over here is another sarcomere. From right here to over here is another sarcomere. But we'll go ahead and just look at this guy here. And sarcomeres are important because these are the actual contractile elements. Contraction means to shorten. So these are the actual units when you contract a muscle, when you shorten a muscle, these are the actual muscles that are going to contract. And they're going to shorten. Okay, so that sarcomere is going to shorten. That sarcomere is going to shorten. That sarcomere is going to shorten. And by doing that, the entire muscle shortens. And that's what a contraction is. the muscle shortens and it pulls on bones, pulls on tendons, pulls on bones. If that occurs across a joint, then that causes movement. So you need this contraction to pull on the bones across a joint to cause movement. So specifically, you can see that there's a whole lot of, individual stuff going on here. These Z discs, H zones, A bands, I bands, M lines, all that kind of stuff. And, you don't necessarily need to know all that, but what I do want you to know is that a sarcomere runs from what we call these Z disc to Z disc. So this is a sarcomere right there. Okay. Now let's take a look within this sarcomere. And specifically, you can see these dark red bands right there. You have these dark red bands, and then you have these thinner blue bands. And that's what we're looking at down here. These thick red bands, those are these guys right here. These are called thick filaments or myosin filaments. That's something that you need to know. These little blue bands are called thin filaments or actin filaments. That's also something that you need to know. And just to give you a preview, what's going to happen is these thick filaments are going to grab on to the thin filaments and it's going to ratchet them in. Okay, it's going to cause them to move on in, to overlap, and by doing so, the entire sarcomere shortens, but we'll talk about how that happens mechanistically. Yeah, that's basically what this is saying. During contraction, thick filaments form attachments called cross bridges with thin filaments, and that's what's going on here. We have a thick... Oh, come on. I don't know how you bring that. Shoot. Don't know how I get my color back. Figure it out. Okay, so where was I? Thick filaments form cross bridges with thin filaments. So here's a thick filament, and what this thick filament is doing is it's attaching itself to this thin filament. And when it does that, that forms something called a cross bridge. This is a cross bridge here. So for contraction to occur, all of these... How did that... Weird. Get back there. So for contraction to occur, what happens is these thick filaments need to make contact. They need to grab onto these thin filaments. They need to form what we call these cross bridges right there. That's the first step of muscle contraction. Because then what's going to happen is these thick filaments are going to pivot and they're going to move this entire thin filament inwards. They're going to move it in, move it in, move it in, move it in. And by doing so, you're shortening the sarcomere. And this happens at all the sarcomeres. This one shortens, and collectively, the entire muscle shortens because of it. I like this diagram here because it gives you kind of an explanation of what's going on, a visual. This is a myofibril here. That's a myofibril. Here's one sarcomere. Here's one. There's another over there. And here we have, like traditional red. There we go. Here we have our thick filament. Here we have our thin filaments. And what you can see is that this is a relaxed muscle. In a relaxed muscle, the sarcomeres, they're relatively stretched out. Okay? What happens when muscle contracts is the sarcomeres shorten. You can see that this sarcomere shortens. They shorten here. And in fully contracted muscles, the sarcomeres shorten even further. So here's a relaxed muscle up here, and there's the length, and this is a contracted muscle right there. And you can see how much shorter the contracted muscle is. Well, the reason this is happening is because, again, these thick filaments are grabbing onto the thin filaments and they're pulling them inwards. They increase the overlap. So here you can see relaxed muscle, but then you can see here in a contracting muscle, these thin filaments are pulled inward a little bit. And in a fully contracted muscle, they're pulled in even further. Okay, so there's a lot more overlap between the filaments in here because they're fully contracted. Again, this is just a visual of all of this, but these are all the myofibrils in there. And here's a sarcomere. There's another one. There's another one. And what happens is when a skeletal muscle cell is excited, when a motor neuron comes down and electrically excites, that's a lightning bolt, electrically excites the skeletal muscle cell, ultimately it's going to cause all of these sarcomeres to shorten. And by doing that, the entire muscle shortens because of it. The entire muscle shortens. That force is applied to the tendon, which is applied to the origin and insertion. And we know that the origin, is the origin going to move? Nope, the origin remains stationary, so it's resisting that pulling force there. But instead what's going to happen is all that force is going to go to the insertion. Because this occurs across a joint, it allows that force to move the bone and actually cause the action, which in this case is elbow flexion. Okay, so this is a skeletal muscle histology. That's one muscle cell. That's another muscle cell. And what I want to point out is that within these muscle cells, the myofibrils are running in this direction. Okay, they're running in this direction, but what you might notice is that they have these striations that are running in the opposite direction. They're running at a 90-degree angle to the skeletal muscle cell itself. So skeletal muscle is striated. And basically what's causing these striations is the alignment of the sarcomeres. So all of these sarcomeres align together and they form these striations here. But keep in mind that the myofibrils are running this direction. Yeah, so specifically, with what happens with these striations is that all of these thick filaments line up with each other from all the myofibrils. And those are all the thick filaments. Those are the thick filaments. And then these gaps in between them, where there's only thin filaments, that causes these light bands that are in between them. So together they form those striations. Okay, so there's some specific organelles that we need to talk about in skeletal muscle. Skeletal muscle has some highly modified organelles that are found within their cells. And one of those is a myofibril. Okay, these myofibrils are organelles. They're highly modified. They're made just for skeletal muscle, or for muscle. Okay, and we've already talked about those, so I'm going to continue on. Let's talk about the composition, though, of these myofibrils. These myofibrils have thick filaments and thin filaments. This is our thick filament right here. And let's see, each myosin molecule. And basically, these thick filaments are made up of individual myosin molecules. This is a myosin molecule right here. And you can see that the thick filament is just made up of a whole bunch of these packed together. So we have, for each myosin filament, we have a tail. Okay, right there. We have a hinge region, the flexible hinge region, so it's going to be able to pivot. And we have heads. These are the heads of the myosin molecules, and these are important. On these heads, we have two different things. We have an ATP binding site. Skeletal muscle contraction requires ATP. Well, that's where ATP is going to bind to allow skeletal muscle contraction to occur. Another site that we have on here is an actin binding site. These myosin heads, these thick filaments, need to make contact with the thin filaments up here. They need to reach up and grab onto them. How are they going to do that? Well, these actin binding sites are going to reach up and grab onto the thin filaments. So that's why they're important. Yeah, I can come back to this. Un momento. So those are the thick filaments, and then these represent the thin filaments down here. The thin filaments have some stuff going on with them, okay? So we're going to go through that. First of all, the thin filaments are composed primarily of the protein actin. All of these little blueberry-like things here, these are actin molecules. And by the way, this myosin molecule, these are all proteins as well. So when you eat a steak, for instance, or hamburger or something like that, high in protein, right? Well, it's high in protein because these thick filaments and these thin filaments are made up of protein. This is the actual protein that you're consuming when you eat a steak. So yeah, these thin filaments are composed primarily of these actin molecules. And these actin molecules, if we take a close look at them, they have what are called these active sites. And this is where myosin molecules are actually going to attach to. So if you have your thick myosin molecule down there, there's the head right there. For this attachment to occur, the myosin head has to attach to this active site on the actin molecule. So this is where that attachment is going to occur. So that's an important component of the actin molecule, or those binding sites for myosin. We have another structure here that's associated with the thin filament, and this is called tropomyosin. Tropomyosin is this rod-shaped protein that wraps around the actin molecule. And what it's doing in this particular location is, you might see this, but it's running right over the top of those myosin binding sites. So it's blocking those myosin binding sites. So in a relaxed muscle, this is what it's going to look like. You're going to have this tropomyosin molecule here wrapping around the actin filaments, and specifically it's blocking these myosin binding sites. So we can't form, myosin cannot reach up and grab these actin filaments. Lastly, the last thing that I want to talk about here on these actin filaments are called troponin molecules. And these little yellow spheres here are troponin molecules. There's 3 of them, and these are the 3 that are represented: TNI, TNC, and TNT. And there's only one of these that I want you guys to be concerned about, and that's this TNC one. And we'll say, one of these is the TNC one there. What TNC does is it binds calcium ions. Okay? And that's going to be critical for skeletal muscle contraction. Is that this TNC molecule is going to bind a calcium ion. Now let me come back to this. Specifically, what's going to happen when a neuron comes down and innervates an individual skeletal muscle cell. This is an individual skeletal muscle cell. Those are the striations right there. Ultimately, what it does is it causes the release of calcium within the cell. And that's what this is representing here, calcium within the cell. Calcium is going to bind to the troponin molecule. And when it does, that troponin molecule rolls out of the way, and now these myosin binding sites are exposed. That allows the myosin head to attach to the actin molecule and form what we call a cross bridge. And that is required for skeletal muscle contraction. We need these cross bridges to occur, and then what's going to happen is it's going to ratchet it forward, and it's going to actually cause contraction. Okay, but this is putting it all together here. Okay. Another highly modified organelle within a skeletal muscle cell is something called the sarcoplasmic reticulum. And this, again, is our skeletal muscle cell. These are our myofibrils. And what the sarcoplasmic reticulum is it's this web-like structure here that's surrounding each of the myofibrils. The reason the sarcoplasmic reticulum is important, maybe you remember this from cell biology, but the sarcoplasmic reticulum stores calcium. And what's going to happen is when a neuron comes down here and stimulates the muscle, the sarcoplasmic reticulum is going to release this calcium. Then the calcium, as you guys know, it binds to that troponin molecule and enables skeletal muscle contraction to occur. So the sarcoplasmic reticulum stores calcium, and the release of calcium is required for skeletal muscle contraction. And lastly, another structure that we have to know is something called the T-tubule. So again, here's our skeletal muscle cell. And this right here is the plasma membrane of the cell. The plasma membrane of a skeletal muscle cell is called the sarcolemma. Well, when a neuron comes down here and electrically stimulates the skeletal muscle cell, we want that electrical stimulation to get deep within the skeletal muscle cell. We just don't want it to cruise along the plasma membrane. So the structures that allow for that are called these T-tubules. These are invaginations or extensions of the plasma membrane. And they have this honeycomb appearance. So here's the plasma membrane that comes down, and it invaginates, it works its way in, and it cross-sections. I don't know how to... Well, there it is there. It makes a cross-section through the skeletal muscle cell, right there, right there, right there, right there. And what that does is it allows the electricity to travel from the plasma membrane and to get deep within the skeletal muscle cell. And because we need that signal to get deep within the cell if we're going to have the whole cell contract. So those are the T-tubules. And some structures that, when we actually look at the skeletal muscle contraction in the next episode, that action potential is going to travel down the T-tubule here, and by doing so, it's going to electrically stimulate the sarcoplasmic reticulum and cause them to release calcium inside the cell, which is going to allow for contraction to occur. Okay, and we can